Simon's had a very distinguished career, as you've heard before probably. He's a Fellow of the Royal Society. He holds the chair of evolutionary paleobiology here at Cambridge. And one of the things he did, amongst the many other lectures and awards that he's received, was he gave the Royal Institution lectures on television in 1996. Very prestigious lectures which reach out to a lot of people. He's going to talk, as you see, about the Cambrian explosion. Remarkable period when life expanded very quickly. And I've just come back yesterday from Iceland, where I work, and you can stand in the middle of Iceland, and there's a real grandeur to it, I think, because all you see is rocks and volcanoes, and there's no trees, there's no animals, there's no birds, there's no flowers, not even any grass in the interior. If you look really hard, you find some lichens. If you go to the hot pools where the volcanoes are bubbling away, you find a lot of algae. And I guess the world was a little bit like that before the Cambrian. So how we came to be where we are now, over to Simon. Thank you very much.
Thank you, Bob. Thank you. Yes, well, here I am again. Sorry about that. I'll try and be succinct. Given the theme of your meeting, and especially the slight leaps we're making from the fascinating material I just heard, along with the rest of you on bioethics, it might seem that there's a slightly jagged progress, and there's nothing wrong with that, of course, because these are very diverse areas, and some have immediate ethical implications. Others might seem to be quite remote, and that would include, it would seem, the Cambrian explosion. I guess the reason, though, of course, I'm far too polite to ask. I always say yes when I'm offered large sums of... No. I'll rephrase that one. With respect to the Cambrian explosion in this sort of context is twofold, one of which I'll come back to right at the end of the talk, but in fact is largely echoing what I was trying to explain in great speed earlier this afternoon. But the other one is that as an evolutionary event, it has attracted attention from so-called creationists. There was a great furore, some of you may have heard about or even read the article in the Proceedings of the Biological Society of Washington, frankly a rather lamentable paper, arguing that there were aspects of the evolutionary process in the Cambrian which would imperil the Darwinian viewpoint, which I think is simply not true at all. Nevertheless, it is scientifically, and apart from anything else that's worth talking about, extremely interesting. It's also pretty controversial, as I'll try and explain, and it's very much work in progress. As I'll try and explain a little bit of where I do some of my work. There are exciting discoveries happening every year, so it's a very sort of topical thing. So there is a background to all of this, which will be, even if you're not a biologist or if you are and are far removed from evolution and paleontology, I hope will have some sort of relevance to the sorts of things we're going to explain. And halfway through there's another take-home message which I'll try and remember to mention. So one thing which of course is well known in its own way, but there's nothing new about the Cambrian explosion. Of course it's familiar to the majority of us because Darwin famously agonised about it. And Darwin, I have actually sort of some ambiguity about terms of sort of one of the most brilliant scientists who lived. Of that there is no doubt at all. But there's sometimes in some of his writing, and more particularly in his correspondence, a slightly sort of Eeyorish, sort of, oh, everything's hopeless. Oh, I think my enemies are everywhere. Oh, Huxley held me on this one at the moment, etc. And then on the other hand, he would sort of, swing out of this, and he'd be sort of doing little hops around the room, saying we're right. So he sort of rather oscillates, between one and the other. But in the gloomy aspects, I think it's chapter 9, with regard to the problem of why apparently when nothing happened and then everything happened, was this consistent with what he regarded as his theory? And to a certain extent, that problem remains with us. So I don't think it's in any way as serious as Darwin might have imagined. But more particularly, this is actually, if I hope it correctly, the bust of William Buckland, a most remarkable individual, quite astonishing. He didn't get on with Darwin. Darwin disliked Buckland quite extensively, and I think largely because Buckland was a sort of unreformed Georgian. That is, he was, a sort of rather sort of, loud-mouthed and probably drank a bit too much possibly, and he was also famous along with his son Frank Buckland for more or less eating his way through the animal kingdom. There was nothing they didn't try. But he was far ahead of his time in areas to do with what we call taphonomy, that is the way fossils actually form, far ahead of his time in the way in which tracks and trails and other indications of what we now would describe as ethology, behaviour, are part of the paleontological input. He was a very, very original man indeed. And in his Bridgewater treatise, which of course in a certain sense echoes a Palean agenda, and that was certainly true to an extent with Buckland, because earlier in his career he'd subscribed fairly seriously to a diluvial hypothesis connected to some sort of flood, or some sort of description. You can see in his famous stratigraphic diagram that there's something wrong with our fossil record. It basically gives up at what we now know was 500 million years ago, approximately. And when you go into the older rocks, then you find very, very little. And as Darwin articulated, but of course Buckland also understood with the early geologists who were active effectively from about after the defeat of Napoleon, roughly 1815 to 1830. This is something which was staring him in the face, so nobody could miss it at all. And I've shown this diagram in one context, but it's also useful in another way because, first of all, we know that the Earth is more than 4.5 billion years old, and the evidence to that, especially from meteorites, is absolutely compelling. Tactfully here, the origin of life has been left out, though Darwin, in his own writings, reasonably, though it turns out probably erroneously, thought that the rock record might well encapsulate the origin of life. It was not at all an impossible assumption from his knowledge at that stage, but that is less likely to be true now. Probably that's all been destroyed, mostly during the major bombardment episodes which preceded the establishment of an effective stratigraphic record, which begins at about 3.8 billion years. But a very curious thing, which has a bearing perhaps on the way Earth-like planets do or do not evolve, and this is all work in progress, is that we do have a sort of fossil record which goes very far back, and this is oscillating somewhat, but there are pretty bona fide fossils from 3.5 billion years, and there's been a lot of discussion about the earlier stuff, but it looks as if it's actually hardening up again to suggest there is reliable evidence back to 3.8. This is microbial material. There are certain key steps which we have a little bit of information on, but there's an awful lot we don't know about. There's one group of microbes known as the cyanobacteria, which actually tend to fossilise rather more readily for various reasons. But it's a very sporadic record, but nevertheless it is there. But the extraordinary thing, and Bob and I have another colleague in earth sciences here in Cambridge, a guy called Nick Butterfield, very entertaining, very intelligent individual, and he's done a lot of work on this interval. And he, along with some other people, it sort of dawned on them in the last few years that there's something very strange going on here, because as he says himself, nothing happens. The rates of evolution for much of Earth history are absolutely glacial. It's incredibly tedious. Nothing happens. You can have, now of course, we know we're dealing with an incomplete fossil record, naturally. We also know we're dealing with forms which may not reflect biochemical complexity. But so far as we have identifiable structures, you can see a microbial fossil separated by 800 million years, and it has not changed. It's very strange. Well, it's apparently strange. Most likely, there's a great deal of discussion about this, most likely it is simply that the environmental conditions, possibly surface temperatures, possibly oxygen as well, were simply not conducive to the sorts of rates of evolution which we associate with the Cambrian explosion. It may be, therefore, that the environmental changes are an important part of the evolutionary story. That shouldn't surprise any of us. But even so, this sort of Darwinian lethargy here is really quite striking. So the first question we have to say is that, given that the fossil record is not complete, that's clear to everybody, and given that there are lots of gaps, and nobody's disputing that, you've got to say, well, what on earth did the first animal look like? There we are. This is JBS Haldane. Again, along with the Haldanes and the Mitchisons others, and there's a fair scattering of that family still in Cambridge. A very remarkable man, like his father. But Haldane is very famous for his quote about, what impresses him about God's action in the world, and it is, his abundance of beetles. But another quote of Haldane's was there anything which would make him give up his belief in evolution? And apparently, I got this story from Mike Benton. I've not verified the source itself, I'm afraid. I have no reason to doubt Mike's absolutely correct. He said, well, if somebody was to find a rabbit in the Ordovician, so effectively, if we were to find some advanced form way back in the Precambrian, then things would not make sense. Now, in point of fact, I think it's now becoming fairly clear where the answer is going to end up. But there is a major dichotomy of opinion, and I think this is actually quite important for understanding some aspects about evolution. Now, at first sight, it might make my position more questionable, possibly even less tenable. As to, basically, is the Cambrian explosion real? And as another individual, Bruce Runniger, has said some years ago, are we looking at an explosion of life, or are we looking at an explosion of fossils? In other words, if there are changes in the potentiality for fossils to form, and the most obvious example, as I'll explain in a moment, would be the development of skeletons, because once you've got a skeleton, that's a pretty tough object, and it basically it behaves as a sedimentary particle. It enters the rock record without any trouble. Or is it, if it's not an explosion of fossils, in other words, is it what we would call a taphonomic threshold, because taphonomy is basically saying what is a post-mortem history of things which, for the most part, especially as I explained in the Burgess Shale, are less likely to be preserved, versus is it actually genuine explosion of life? And this has actually been very controversial for two reasons. The first is that Darwin's dilemma, as we could call it, might be explained simply because we've not interpreted the fossil record correctly. In other words, there may be a long, long history which ancient professors in ancient universities have turned their back to, refusing to accept the evidence from thrusting young, brilliant graduates who show the evidence before them, but these people are so hidebound that they are unable to change the world picture. No, it's not like that, I don't think. But there are two lines of evidence which appear to come together to suggest that maybe Darwin's dilemma was simply that we were overlooking a long, long term history, so that eventually when the Cambrian explosion happened, it was a cumulative product of many previous evolutionary processes. And the evidence came from two lines. The first was that going back more than 1 billion years, the Cambrian explosion effectively starting at about 550 million years ago, we do have various structures. These are ones actually from India. This is on the front cover of Science, so you wouldn't know it, which were claimed to be primitive burrows, that is, trace fossils. And this was got a great deal of attention because, after all, they were highly sporadic. But if there are soft-bodied creatures which may not have skeletons, they can still move through the sediment and leave behind them some imprint. I mean, of course, a dinosaur trackway would be the most obvious equivalent the majority of you would be familiar with. And in this particular case, in fact, I've seen some very good replicas of this material in the Describers lab in Germany. This is a work of Dolf Silak, who's a very brilliant paleontologist. I don't think these things are genuine biogenic activity at all. I think it's actually very unlikely. But this actually appeared to combine very nicely with a separate line of evidence, which actually turns out to be important in other sorts of contexts, because, of course, a vital part of the evolutionary explanation depends on molecular data. And one key aspect is that to the very first approximation, here I'm going to simplify in a way which will embarrass some people tremendously, because the substitution, say, in a protein is approximately random, the reason why, for instance, our hemoglobin is identical to that of a chimpanzee, and I think it's one difference with a gorilla, something like that, you know, it doesn't really matter too much, it's because, of course, we're very closely related. But correspondingly, if you compare our hemoglobin similarity to a cow, actually there are a few differences, not that enormous, and you go a little bit further afield, and then those differences become increasingly proportional, because our common ancestor lived proportionally earlier and earlier and earlier. And it turns out, rather oddly, that if you plot the rate of substitution through geological time, and this is fraught with problems, it's often surprisingly constant, which means that if you know how different the molecules are in two living organisms, you can infer when they must have split apart in the geological past. This is the so-called molecular clock. And using the molecular clock was consistently suggesting that animals must have evolved more than 1 billion years ago. And this didn't make any sense to me, because I've been working on this area of the fossil record for some years, and things were actually getting quite heated. So I thought that we ought to go to neutral territory. We should have some place which, you know what science is like. You may have noticed they end up in, Bermuda was mentioned earlier. Did you see that? Yes, it's odd they don't go to Skegness too frequently. So I decided to go to Venice. It's a city which I'm enormously fond of. And I went a day or two early, in fact, and I took the number 1 Vaporetto out to the Lido, and I knew the opposition team would be arriving, and we'd all be going out to one of the big hotels there, and we're going to have one of these sort of special things and try and thrash out our differences. In any case, I looked across the lagoon. I had a pair of binoculars given to me by my wife Zoe, who will be coming to the dinner, if you may or may not be pleased to know. And this is what I saw coming towards me. Here with the opposition. Oh yes, that's right. they were taking no chances on this particular occasion. That guy there, by the way, is a claydist as well. That's another story. But But it so happened, in fact, I had a rather large research grant, which is not like me at all. And my own team was also coming, but they were coming from another corner of the lagoon, and I thought you'd like to see a photograph. Here they come. I think we did, this particular one, we did actually pretty well. So these discussions have been toing and froing, but I think in the last couple of years, by and large, it's becoming less and less likely. In part, it's because I think we've rather improved our knowledge of the fossil record, but more significantly, in fact, is that the early attempts at the molecular clocks are now seen to have been a little bit simplistic, and there are ways in which you can refine the molecular information. And there's especially a very talented scientist in the States called Kevin Peterson, who's done some work with Nick Butfield in Cambridge. And he and a number of other people, I think, have made enormous progress in refining and actually correlating the molecular data with the paleontological data. And I think things are actually a great deal more comfortable than they might have been some years ago. Things do seem to be making sense. Now, one of the difficulties with the understanding of this evolutionary event, which I think is genuinely interesting and genuinely important, is that just before the major appearance of skeletons, which is the thing Darwin recognised, but as we now understand is very much part of a larger event, it's not only skeletons, is that there are these extraordinary biotas which are broadly known as the Ediacaran assemblages. Ediacara is a set of hills in the Flinders Ranges in South Australia, but these faunas are global. There are wonderful examples in Newfoundland, elsewhere in Canada, in the White Sea area, some in China and so forth. But the extraordinary thing about them is that they are, for all intents and purposes, completely soft-bodied. There are no skeletons there at all, but they're abundant. They occur all over the place. So how come soft-bodied creatures are preserved so readily? The other thing is that they are sort of similar, and so we have these things like fronds. I'll come back to those in a moment. And we also have things which might be sort of worms. But I must point out immediately that we have got the obligatory jellyfish. Now, when you're depicting an archaic seascape, I cannot emphasize too strongly how important it is. You must have jellyfish. Very, very important indeed. It's the same thing if you look at a diorama of a dinosaur tearing another one apart, typically, then there's always a volcano in the background. In fact, you'll probably notice they actually use the same volcano in all dinosaur dioramas. Sometimes the smoke goes that way, sometimes it goes that way. It doesn't matter too much. In point of fact, it's most unlikely there are any jellyfish there at all for various reasons. They're probably actually somewhat more advanced than one might imagine. They've probably actually evolved later. But nevertheless, these are real assemblages. They are real faunas. In addition to this, there are extraordinary discoveries, principally from China, which I think are just worth mentioning because, again, so far as the fossil record is concerned, I mean, I used to be very keen collector as a schoolboy and all that sort of business, doing dangerous things with my hammer. But again and again, we see that in the sedimentary record, the fidelity of preservation is little short of astonishing. But one of the remarkable things is from these deposits in China have been recovered. They're quite small, they're around about 0.5 millimetre or so. These, I'm not sure if they make a lot of sense to you, but these are individual cells, and this is an embryo. So this is an embryo of some organism, and the reason it's been preserved is because phosphatic minerals, they're actually very similar to the minerals you find in your bone, they're basically hydroxyapatites, one sort and the other, have what has been called a Medusa effect, a very rapid fossilization from something which you would think would be so intangible as to have no chance of entering the fossil record. And these things have generated an enormous lot of discussion for various reasons, one which I'll come into in just a moment. But in a funny sort of way, they actually haven't told us much more than we really wanted to know. The most curious thing is that by and large they're not known to develop much before what we call a 64 cell stage. Not quite sure what they evolved into. I mean, for instance, some of them, for all we know, may have been the early embryos of that, or they may have been the early embryos of that, but we're not actually sure, unfortunately. But the take-home message at the moment, and there was a couple of papers, one in Science, one in Nature last year, where they were using an extremely sophisticated technology using synchrotron radiation, where they're able to investigate the interior of these embryos, and remarkable imaging. But they do appear to be basically from primitive animals, and frankly, that's almost all you can say. So in a certain way, remarkable as they are, they haven't quite filled in all the gaps which we would like. There is an additional problem, though. This is actually the main locality where they come from. This is in Guizhou province in southern China. A little bit of geology for those who like that sort of thing. This is a Russian for scale, if that helps. This is actually an ancient land surface here. This is limestone which was exposed above the sea, was formed to sort of what we call karst then it was reburied in Morsey there, and most of the embryos actually come from this locality here. And for some time, they were thought to be very much older than the Cambrian, but in fact the radiometric dates have been revised successfully. It's pretty clear that where this chap's foot is only about 10 million years before the Cambrian explosion itself. But amongst the other things which have come out of this are such structures as these, and I certainly don't want to I don't want to be too rude about this. This has got into science and a big hoo-ha about it. It too is a phosphatic microfossil, but it has been reconstructed as a miniature animal with things like mouths and body cavities here. And I mean, I could use some fairly objectionable language, and I may in a moment if I don't control myself, but this is, frankly, it's total rubbish. This is an artifact it's inorganic. And it actually does have a slight take-home message, even within the bona fide Ediacaran fossils. And ultimately, for what we're trying to do with science, it's the way you actually recognise patterns, how you make sense of things which may not be familiar. And it would also be very important, perhaps in the future, because if we're engaged in serious exobiology, and we're looking at extraterrestrial planets which we cannot directly visit ourselves, and we're using remote sensory systems, and you have an organised structure in front of you, can you be sure whether it's organic or not? So there are backgrounds to this, but there's a whole psychology and a lot of activity, and really this idea that these things are tiny, tiny animals, I'm afraid it has won no supporters outside the people who originally described it. So there are some problems there. But there are also problems with a number of these organisms. I'll just give you a couple of examples. This is a creature which is found principally in the White Sea area of Russia. It's a worm-like animal. It typically grows around about 3 or 4, 5 centimetres in length. You can see it's got a segmentation to it. But the extraordinary thing is that in the rock itself, you can see that it forms a series of impressions behind it. So what on earth is going on here? It's got an axial division. I've got a backbone. It's got transverse divisions. I've got a rib cage. It's got a front. I've got a head. So it's something like an animal. But why is it investing all this effort making these impressions into the underlying mud? And most likely, I think, is that probably it's taking nutrients in. But if it's taking nutrients in through its undersurface, and that's quite all right, that's a way of doing it, which is quite acceptable, that suggests it doesn't have a gut. So if it doesn't have a gut, then it's not the sort of animal we're familiar with. So you can see that there are problems with that. And then if we look in detail at this particular animal here, it's called York, if you're interested. Again, it's sort of, it's familiar in a way. But in other ways, it doesn't quite fit into what we expect. Now, I have some ideas where this thing might fit in, but it's quite, especially if you're trying to teach science, the balance between, obviously you have to accept most of what you're taught, otherwise you can go mad. But on the other hand, having an open enough imagination to realise that sometimes ideas have just got to be thrown away. And in particular, when it comes to the fossil record, and perhaps this is the time of the take-home message, is, in my view, the fossil record is essential if you're going to study evolution. Not everybody should be a paleontologist, but it does provide unique historical data on transformation which are simply unavailable from any other line of evidence. That doesn't mean other lines of evidence are important, they're equally vital. But these things here are real data. My suspicion is that these are basically what we call in the trade stem group triplobasts. I think they are probably the most primitive animals, but they are very difficult to understand. And even worse is that there's a whole variety of other fossils which look vaguely animal. This is an animal called pteridinium. It's best known from Namibia, but it occurs in a number of other places. Again, you can see it's quite large. Some of these jokers can grow up to 1 metre in length. You can't miss them. It's got this long set of divisions. It actually had a trifoliate cross-section, and again a series of cross divisions here. But is this an animal? It probably isn't. Dolf Zeilacher, the person who described those possible Indian trace fossils more than 1 billion years ago, has also been extremely influential in many positive ways. And at one stage he tried to persuade the world that all of these things are a completely separate evolutionary experiment. Things he called the Vendozoans. They look like animals, but actually they are protistans, which are, in a sense, quasi-animal-like. I think, in fact, he's half right, half wrong. I'm fairly sure that some things genuinely are animals, but these sorts of large, macroscopic creatures are evolving at about the same time. And for all we know, they may have even been using some of the same genes, because there are various genes involved in animal organisation which are responsible for the various ways in which you can divide the body up. And it's quite possible that these have been inherited from a common ancestor. But once again, these things in one way, are incredibly frustrating. They're preserved in sandstone. You can cut a cross-section through them and you learn almost nothing. There really is a limit to one's knowledge, and again, that's something scientists don't generally like to talk about, but we live in a surprisingly cloudy world. Then, of course, we have the invention of the skeletons. They're the things which are obvious to everybody. This must be a French slide. And the skeletons have for a long time been the focus of attention as to what might have actually initiated the Cambrian explosion. They're certainly part of the argument, but it's now clear, as I hope will become obvious in the next few slides, that in point of fact, in Cambrian assemblages, probably no more than 5 to 10% of animal species possessed skeletons. The other things were soft-bodied. Skeletons seem to have principally evolved as a protective mechanism. They may be linked to changes in the chemistry of the ocean, that's highly debatable, but nevertheless, they are our most useful source of evidence because they're abundant and readily fossilized by definition. But in point of fact, in terms of the totality of our understanding, they are insignificant. The other thing which is very interesting is that, however controversial the earlier so-called trace fossils might be, there's absolutely no doubt that genuine trace fossils, such as this one here from the Cambrian, which is made by some sort of perhaps mollusk-like creature, probably moving within the sediment, their diversification, for all intents and purposes, is directly parallel with the diversification of the body fossils. And because we have reason to think that the majority of these so-called trace fossils, the burrows and trackways, the science of what's sometimes called ichnology, after the Greek word ichnos, marking, the fact that that's parallel with the body fossils of the hard parts, to my way of thinking, suggests that, again, this is a genuine biological event. And the trace fossils are, for most parts, are proxy for what is a soft-bodied material, which in normal circumstances hardly ever survives. But sometimes it does. And I'll say a little bit about two of the most important localities, because curiously in the lower and the middle Cambrian, so here we're dealing with a time period which for our purposes is effectively from around about 525 million years ago to around about 505 million years ago. So we're only talking about around about 20 million years. We have a series of deposits found in many parts of the world which have soft part preservation, of which the Burgess Shale is probably still the best known. This was discovered by Charles Walcott. Some of you may know the story, some of you may know Stephen Jay Gould's book, Wonderful Life. And they received permission to reopen these quarries in the 60s, and it's this man here, Harry Whittington, who was asked to lead the paleobiology excavations, and by great good fortune, he was in Harvard at the time. He was very settled. His wife Dorothy, who died some years ago, Harry's still doing fine. He's about 93 now. He's still writing books and publishing papers on trilobites. Then got the invitation to come to Cambridge, and that enabled myself and another chap called Derek Briggs to actually become PhD students with Harry. So that I can then instantly explain this. By the way, this is not me, if anyone is sort of interested in that. So this provided an impetus for the reinvestigation of the Burgess Shale to begin with, and since then things have, in a manner of speaking, snowballed. So what I'm going to do now is just take you at high speed as usual through some of the main animal groups, and quite a lot of this, I'm afraid to say, is based on my work, which means that those are my many enemies, especially some dear boy in London, obviously would disagree with much of this. So there's a health warning coming out there. And also there's a great deal of controversy. One is still dealing with quite a sporadic fossil record. But what I think is important, I mean the take-home message, for want of a better term, is that I'll try and persuade you that the fossils we have are genuinely informative about the construction of body plans. And similarly, you have to throw away your assumptions about what the ancestral types look like unless you use the fossil record. Because zoology textbooks in particular are stuffed full of hypothetical archetypal reconstructions of what the common ancestor of two disparate phyla, that is body plans. And I think most of those are simply fictions. They are totally imaginary. But it's not easy. And this is the first example. I don't even, I'm not going to ask you, who knows what this is. These are sea gooseberries. These are ctenophores, extremely beautiful creatures. Wholly marine, largely pelagic. And they have an incredibly uniform body plan. Effectively, they have a gelatinous body And I hope you can see these rows along here. These are rows of fused cilia which beat in metachronal rhythm and propel the animal through the seawater. And they're also extremely effective predators. These are of major interest to fisheries and so forth. And the interesting thing about them is that they have a fantastically stable body plan. They've all got 8 rows of these fused cilia. You've seen one ctenophore, you've seen them all. So where do they come from? Well, we know that they're sort of related to another big group, which I'll come to in a moment, which we call the cnidarians, which includes such things as the corals. But in point of fact, they're not as close as people thought. Well, there's a great deal to play for here, of course. To begin with, we do find what seem to be equivalents, for instance, in the Burgess shell. This is a fossil ctenophore. It's somewhat bigger than some of the living forms, but interesting, it has many more of these so-called comb rows. Comb, again, the Greek tini, if I understand that correctly. But this thing seems to be an identifiable ctenophore. But that's sort of helpful, but that doesn't really help us very much at all. Now, this is a big jump, and we may be completely wrong in that area, but in addition to the Burgess shale, there are 2 other localities, or 2 other regions, which have turned out to be especially productive. One is in Greenland, and I'll come to that in a moment. The other one is actually in China, where I've done quite a lot over the last 10 years. And a couple of years ago, rather, my friend Dagan Xu, I work with a group in Northwest University in Xi'an, he sent me some photographs, and we got this paper out in Science, I think earlier this year, if I remember correctly. And these are very interesting fossils. I won't go into all the gory details, but there are really 3 things to take home. First of all, this is Burgess shale type preservation from China. It's a little bit older than the Burgess shale itself. It's beautiful preservation. Second of all, it actually has a frond-like shape, which is remarkably reminiscent of some of those ediacaran forms. So maybe there's a connection with what have otherwise been rather enigmatic ediacaran forms. Again, not everybody buys this story at all. The third thing, though, excuse me, is that if you look closely at these individual branches, and it's not too clear even here, these branches here, even though this animal itself was completely sessile, we suspect were ciliated, and we speculate that effectively before the ctenophore learnt to swim, it was a benthic animal living in the seafloor with the cilia being used for some completely different function. Now, there's no reason why you should buy that argument when I give it to you in 2 minutes 34 seconds. There are a whole series of other steps to be made, a whole set of different sorts of argument. But I think there's some merit in this hypothesis, and if it's one worth exploring, it is again the possibility that when you're trying to understand the transformation of body plans to produce what we regard as stabilised phyla, whatever that's meant to be, then the likelihood is that the precursors are going to look quite different from what you'd expect. And I say, once this begins to sink in, it's really not very complicated at all. But it does mean, and this is certainly an area where I've changed my mind quite radically, because when I first started work on the Burgess shale, I was far more impressed by the sort of sheer strangeness of these things and the difficulty of understanding them. But as our work's progressed, so I think we've been able to make a bit of progress in that area. So possibly, this thing is an ancestral ctenophore, but it doesn't look like one. But we don't worry about it, because the evolution is true. Here's another example. This is actually from the Burgess shale itself. It's much bigger, it's about 30 centimetres in length. Charles Walcott found this. There are only 3 specimens. He never described it. And this one I described more than 10 years ago. Also a frond-like form, strikingly similar to some of those ediacaran forms. And in point of fact, if you look in detail at some of the anatomy in this sort of area here, and I'm not sure it's any clearer in the reconstruction, but that may be something you're slightly more comfortable with, these frond-like structures here, we would suggest in point of fact are closely related to the beginnings of another primitive group known as the cnidarians, which include both the living sea pens, the penatulaceans, but also the corals and the sea anemones, and also those jellyfish, which I mentioned in passing. So here too we seem to have an ediacaran survivor in a Burgess shale setting, so we're allowed to have trilobites and other more primitive arthropods there. So again, it's early days. There are still big jumps in our argument, but all the time, like any sort of science, however erroneous it will turn out to be, obviously what we're trying to do is organise a matrix of understanding. Now, you need to get your binoculars out, of course, to look at this diagram. But what it's trying to show is that there's a geological timescale down here, and we're going across the Cambrian explosion in the column there. These blocks of colour here are the 3 major groups of more advanced animals. And all I need to emphasise at the moment is, and this is where molecular biology has made a profound difference to our understanding of evolution, is that in the last 15 years, the study of molecules from living organisms has suggested evolutionary relationships between various groups of animals, some of which are relatively familiar, mollusks, some of which are relatively obscure, such as nematodes. And it suggested evolutionary relationships between these groups of animals, which traditional zoology regarded as basically impossible. And in fact, it's quite interesting to see how the old guard I can think of one person in particular, are more or less going to their graves, are unable to take on board these new ideas. And it's not that they're stupid, far from it. Of course, they have a great deal of information from other lines of evidence, for instance, embryology. And it's all a question of believing what is possible in evolution, effectively. What is likely to re-evolve? If you think something is unique, it's so difficult to evolve that it could have only evolved once, and then you find it in different groups of animals, you have to say, well, they must be related. But in point of fact, as I was trying to explain with regard to convergence, and I give you many other examples, that I think is a very unwise thing to do at the moment. It's not the way to drive your research program forward. But if we take, for instance, this central group here, which rejoice in the name of Lophotrochozoans, and I'll give you a couple of examples in a moment. If I say that 3 body plans, which all look completely different, one from the other, are all related, then any other evolutionary biologist is quite entitled to say, well, what on earth did it look like then? Come on, show me, show me. Oh, it's got to be real. I don't believe in this rubbish. And that's where I think the Burgess shell faunas are turning out to be quite informative, because we found a variety of very strange-looking animals, which in point of fact, I think, are showing us how to build a body plan. But by no means everything is straightforward. This is a rather sort of difficult to understand photograph, but this is a grasping spines in the front of a very interesting group of animals known as the cheticnaths, or the arrowworms. It's actually having a herring larvae for breakfast. These again are great interest to fisheries, they're very effective predators. The cheticnaths have got a unique body plan. It's totally distinctive. You've seen one cheticnath, you've seen them all. They're very, very standard, very easy to recognise. And even now, even with molecular biology, even with the fossil record, we still are remarkably poorly informed as to where they fit into the system. There's been some progress, as a group in Marseille have actually made some very substantial progress, but actually these things are still very much an evolutionary enigma. So there's still a great deal to play for. So by no means everything is clear-cut, but I think we are a little bit further along than we might have expected. So, to explain how we get there, again, the sort of background is we've also got a Burgess shell locality in Greenland, up in Pearyland. We've been there 3 times. I just thought I'd show you through a few field shots. This is JP Cockfield. You can see all the sea ice there. How much longer that'll be there, of course, is another question. It's a wonderful part of the world to visit. To get there, you're put in by Twin Otter. These are Icelandic pilots, actually brilliant people, superb. Our locality was about 30 kilometres from this locality. This is a little shed which is used by the Sirius Patrol. That's why we called the thing the Sirius Passet Lagerstätte, Lagerstätte being a German word for exceptional fossil locality. So they could put us into this strip here, but we really need to be down at the other end of the valley. So we had some pretty interesting experiences landing an aeroplane on tundra, but we got down safely, fortunately. And then, after being put down, we still were some distance from the locality itself. So my friend John Peel, I'm doing quite a lot of work with him again at the moment, who at that time was in Copenhagen, realising, of course, and being a Greenland expert, there aren't many forests in Greenland, thought it would be a good idea, especially if we had to tow all our fossils many kilometres down the valley, to have these sorts of trolleys here. So we had to move camp several times before we were close enough to the area. And you can probably sort of understand this being a pretty well English expedition. You'll be interested to know that we have most of our claret in there. We have some very good burgundies in there. And John and the other ones are bringing some of the Alsace wines after the back. So that was all fairly satisfactory. And of course the top was made mostly of Mars bars. This again being very much an English expedition. And we had to move camp 3 times. We finally got to a place not only where we could camp reasonably close to the locality, but also somewhere where we could build a new airstrip so we could be taken off safely by the Twin Otters. And I'd asked John here, what it was like. Was it dangerous? He said, yes, pretty dangerous. Have you ever used a gun? No. Right, you might want to learn how to use one of those. Polar bears? Yes, very common, very common. And also he said that apart from that, and the plane crashing and other such things, it's a wonderful place to be, Simon, because we have this sort of brilliant weather. And he went into a very technical description about high pressure areas. And because we're at 83 degrees north, the sun never sets here, the sun just goes round and round the sky like that. So he said, Simon, if you get really keen in this area, you can collect all the time. He was wrong. The weather's awful. And you can still see the trolley there. I interviewed John in the tent. He actually admitted, in fact, the weather in North Greenland can be truly terrible. That actually was the first season. We went back 2 more times, and both those summers were mostly brilliant. And there's something about having sunlight for 6 days continuously, which to the English heart strangely warms one. The actual locality is actually, in a way, a little bit similar to the Burgess Shale. You have limestones here, and this is the locality itself, and you can trace those limestones going across in that area. It's not quite the same as the Burgess Shale, but in the Cambrian it was still part of North America. And the locality in slightly more detail here, this is Taylor Slope here. All the underlying rock is locked in permafrost. You simply can't get it out, so you've just got to collect superficial material. And many of these slabs are shown. This is a slightly chillier day. This is John's ex-wife Mona. Have soft-bodied fossils. And it's a really very simple job. He's a marvellous person to work with, great expedition leader as well. You just find the slabs rock, put them on a layer of sponge, put them in a box and carry them down. And there you go. So what did we find? Well, when we found things we got very excited, but there were also these tedious views to put up with. So again, any of you who've done Arctic or Antarctic work will know that it's a different sort of world there. Most of the fossils there were sort of Burgess Shale-like. Actually, Burgess Shale is rather recurrent. One thing which was a bit of a surprise is this creature here. They're actually called halkyrids. Got it onto the front page of Nature, it's always nice. And the halkyrids are effectively an animal like a slug. This is the front, and then there on the left, this is the posterior there. So what you've got to imagine is something much the size of a large garden slug, except that its dorsal surface is covered with thousands of tiny plates. But much more surprisingly and completely unexpected is there's a whopping great shell at either end. And that's, I'm afraid that's actually upside down, so our anterior is down there, and then the posterior is there. So who would have guessed that we'd find an animal with 2 shells, one on either end? Of course, that begs the question, what were those 2 shells doing? We actually knew about this group for some years beforehand, because when the animal died, then the different plates which were made of calcium carbonate would be dispersed into the sediment, and we'd recognised those for a number of years, but we certainly had no clue about these shells. And then if you look at a close-up, you can see that the skeleton is beautifully imbricated. So that reinforces the idea this might be a protective structure. But of course we have these, oops, sorry, go back one. We have these extraordinary shells. This is the one at the posterior. Now if I cut out that shell from the photograph and sent it to any paleontologist, he would have said that's a brachiopod. I'm sure they would. I should have done that. It was rather silly of not to have done that experiment. Except brachiopods do not live at the ends of slugs, because brachiopods are bivalved creatures, which are also marine, which feed using this internal set of tentacles, which we call the lophophore. That actually is a connection to the lophotrochozoans. Now, molecular biology had told us that this group, the brachiopods, were actually very closely related to the mollusks. They're things like the clams and the snails, and also very closely related to the annelids. I showed you a camera eye in cross-section, but also include the more familiar earthworm. So if you say, look, there's a snail, there's a brachiopod, and there's an earthworm, can you tell me what the common ancestor looks like? You know, what transformation? They would be completely arbitrary. But my sense is that actually the halkyrids may be telling us something in that direction as to what they're actually, what they are related to. And the reason goes as follows. Let's just go back. First of all, in terms of mollusks, this is broadly a slug-like creature, and it doesn't stretch imagination to see this thing transform itself into something similar to a living chiton or a living apicophoran. I mean, there are technicalities there, and I'm very happy to talk about them. I don't think things are as simple as that, but nevertheless, it has a slug-like organisation. What about the brachiopods? Well, don't be silly, but one possibility, of course, is that the halkyrids, of course, have 2 shells which are separated. But early in ontogeny, those 2 shells are, of course, very small, but they're also back-to-back. If those 2 shells were to swing together, they would thereby define a bivalved arrangement, which is a brachiopod. Well, that sounds all highly arbitrary, and it might be, except, of course, we remind ourselves that molecular phylogeny is allowing us to rate a mollusk-like creature to a brachiopod. But when we look at the brachiopod organisation, it's very strange. Nature, when it wants to make a bivalved structure, such as familiar, for instance, in a bivalve mollusk, will take a plate and weaken it along the midline and then fold it together. You see the same thing in the ostracods, for example. You demineralise the central zone. That never happened in the brachiopods. The brachiopods always had 2 separate shells. The ontogeny shows they never had a single shell. And that explains in part why they have such a baroque musculature to articulate those 2 valves together. So that is not quite as way out as it might seem. Again, I'm more than happy to discuss the nitty-gritty about these ideas. I just want to give you a flavour of how you might drive the argument forward. That other group of lophotrochozoans, we call them, include both the earthworm and its marine equivalents, the annelids. And here too I have to skate over a lot of very controversial ground. There's by no means general agreement on that area. One of the curiosities of the annelids is that they have these little projecting bristles made of chitin called the chetae. And it's long been recognised that those actually are identical, let me just go back a couple of slides, to in fact these bristles here. Now that could be convergence, of course, but it's again a straw in the wind that there's a relationship between these particular groups. And in the case of the annelids, there's another Burgess shell animal which is called Wiwaxian. Again, Nick Butterfield's done some very important work in this area. And he would broadly argue, again there's controversy in various corners, he would argue this is effectively a pre-annelid. And we've also argued that this animal, Wiwaxia, although it shows some differences, is also closely related to the Hawaxids. And a couple of very distinguished Danish scientists said that's absolutely rubbish. Conway Morris has finally shot himself in the foot. I could not imagine a more stupid thing to do. So very fortunately we were able to find the intermediate to show to them, and they weren't very pleased about that. In fact, they were so pleased about it they took the data and didn't change their minds and used it, which I have to say is pretty impressive. But these little chaps here are from the Burgess shale, and our argument is that they are intermediates between the Wiwaxids, as they're called, the Wiwaxids being that creature there, and the Halkyrids, and they have a Wiwaxid-like skeleton, but they have little shells at the front there. And this again I think is important. I say we may be completely wrong about all of this, I wouldn't be surprised. But it's the way we're trying to drive the argument forward, because that's a reconstruction of this rather beautiful animal. It's only about just over a centimetre in length. Is that as we find more Burgess shale localities, particular animals which would have looked extremely strange individually are now beginning to fall in place. And again, any of us who do the science on a day-to-day basis is all entirely familiar. But it does suggest that things may look baroque, bizarre and weird, but in point of fact they are beginning to make some sort of sense. So in this context, I think we've made a little bit of progress. Unfortunately, I can't show you the picture, but in point of fact we have here an arthropod, which I'll say a little bit about, but also common in the Cambrian we find these cap shells here, which have always been called monopacophrons neopolina. We have some new specimens from China, and I'm not allowed to say anything about it because it's all it's all slightly under wraps. But all I can say is that you could not be more wrong. Believe me, you could not be more wrong. Within the next great group, which are sometimes called the ecdysozoans, the arthropods, again it's been extremely checkered history, but it's now fairly clear that we have a series of transformations from such things as effectively a soft-bodied worm-like creature, this is called a Shea, to our old friend here, Hallucigenia, which a number of you will know I managed to get I got a really brilliant reconstruction. It was fantastic. It's one of the best papers I'd ever written. The only problem was the animal was upside down, but apart from that it was fine. And then there was this famous animal here, Anomalocaris, or Anomalocaris, which Gordon made a great song and dance about, but it's now very clear that this too is an arthropod. But the take-home message about this, as well as this strange animal here, Opabinia with its 5 eyes, is that this is effectively a series of vignettes into the assembly of the body plan, from something which is effectively soft-bodied to something which is a fully fledged arthropod. And each one of those steps is in its own way unremarkable. Each one of those steps is explicable either in terms of protection or on feeding ecology. And the other thing which is interesting, and I'll make this point I hope again, is that effectively both descendants and ancestors are living side by side. It's a bit like as if you saw in the war room Julius Caesar and Winston Churchill talking together. And that, again, I think is consistent with this being a genuine explosion. And they are reinforced in the ecdysozoan story by a diverse group of organisms which are called the prearchlids, which still live today, or the penis worms as they're also known, which are less diverse today, but these are extremely diverse. And the argument is that something like this, when it came out to move on the sediment, was effectively the precursor of the arthropod, which moved from a peristaltic motion to one using legs in a normal way. And you may wonder how I'm sometimes very well informed about what's going on in the Cambrian. Well, if you look closely, you'll see where all our evidence comes from. So yes, we're not going to rely on fossils. We go there. We go and study them. We have a time machine. We bought it. We can get them on the web. Yes, believe me. And then in this context, and I may need to finish early, subject to Dennis's guidance in this area, I certainly don't want to overrun, is the group which is our group, which I mentioned with regard to the starfish and the human. This is a group called the deuterostomes, and in many ways this is the most interesting, not only because of our own anthropomorphic concern about the group we belong to, but because of the sheer disparity, the sheer difference between especially the echinoderms, epitomised here by this sea urchin. This, by the way, is a parrotfish. What it's going to do is it blows water out of its mouth and topples the sea urchin onto its undefended side and then eats it. So there you go, I thought you'd be interested in that. But the range of forms, vertebrates versus starfish, again, what on earth does a common ancestor look like? I need to press that again. Okay. Well, from Chengjiang, again in China, some years ago, some people described this fossil. You can see the scale bar here. And for those of you in the trade, this looks actually a bit like a group of arthropods. It's got a carapace-like structure there, and it's got the segmented tail out there. And that's what it was described at. But perhaps you can see along the midline, there are these rather odd structures there. And if we look at those slightly more closely, there we are. This is looking into the interior. This is one of the lovely things about Chengjiang in comparison to birdshell. Birdshell is basically a metamorphic rock. It's really hard, tough, and squashed. Chengjiang is much less metamorphosed, much softer, and you can excavate, you can basically dig your way through one side of the animal, and here we see on the interior of this creature, this row of pouch-like structures there. And those pouch-like structures, again, you'll have to take this, I think, on trust, but we are actually interpreting as the precursors of the gill slits. And the gill slits, which of course we find in the extinct echinoderms, we find in another group, the hemichordates, and in our own group, the fish, are the hallmark of the deuterostomes. But they don't look like the sorts of gill slits one would expect. Now, the next slide I'm going to skip over because it's all rather technical, but it just shows basically the sort of drawings we use to interpret these things. It's all rather laborious. And so, that we would argue, and again, this is work in progress. We've got another piece of work underway, both on Chinese and Canadian material at the moment. It is a clue as to what the common ancestor of fish and starfish and other things might have looked like. It didn't look like the things you expected. The echinoderms are particularly peculiar because their diagnostic feature, well, there's several of course, but one of their diagnostic features is they've adopted this five-fold symmetry. All of you who've seen a starfish will have been struck by the fact they typically have 5 arms or mouthfuls. And you think, what are they doing? We don't actually know. The other thing they apparently have done is thrown away their brains. They have convergent compound eyes, oh yes, they've thrown away their brains, that's another story. Very odd indeed. So again, imagining transformation from something which, as Molesworth would say, any fool could recognise, is quite problematic. It's got more problematic because the echinoderms in the geological past include a variety of extinct forms which have generated a huge amount of controversy, principally in the lower Paleozoic. And as you can see, they've got a sort of body here, and then they've got a segmented tail. But actually, if you think back to that fossil I showed you a couple of slides ago, that's got a large body and a segmented tail. Oh, maybe there's something there which is more significant. But the other aspect about the echinoderms is that they make a very particular sort of skeleton, which is instantly recognisable. It's constructed in a particular way. And there's no doubt this is an echinoderm, even though it doesn't look like any sort of starfish. And then when we go to look at other strange sorts of things, there's a lot of information here. We do find these really weird-looking echinoderms. They typically have long tails. Some of them have definite gill slits. In fact, I'm going back to China in December to look at some very exciting discoveries of these from the earliest Cambrian. They're amazing stuff. These things have been very difficult to put into the picture. Some people think they're highly derived, but I think it's now fairly clear that these are part and parcel of transforming yourself from an animal which is moving around actively with gill slits to one which becomes increasingly sessile, ultimately loses its gill slits, and ultimately becomes effectively stationary. And only then do you move into the five-fold symmetry which is characteristic of the echinoderms. So that five-fold thing is an important story. It's an unsolved evolutionary problem, but nevertheless it has nothing to do with the origin of the echinoderms. And we have other sorts of creatures. There's another one from Chengjiang, which we suggest, again, I won't go into all the ghastly details. This is an animal we described a couple of years ago, which we think, again, has a gill slit there. We think that's probably the anus and the mouth that's actually there. And our argument would be that before an echinoderm learned how to make a skeleton, then probably it looks something like this. And then we have, again, reinforcing the message of the Cambrian explosion in terms of the simultaneity of very diverse groups belonging to the same group. Not only do we have what appear to be the earliest deuterostomes, the earliest echinoderms, but rather surprisingly, we also have the first fish. And that too seems consistent with the idea of an explosion. Now, I've got a couple more slides which I'm not going to show. There's another sort of story which we won't worry about, and there's stuff about conodonts with another story, I just want to wrap up quickly. With respect then to the Cambrian explosion, the question still remains what triggered it. After all, as I point out, Darwin's got this for a long time of glacially slow evolution. Then things do seem to move. There's a lot of subtlety and complexity, especially with regard to the Ediacaran assemblages. But we still have no satisfactory explanation of what the trigger might be. There are plenty of ideas. One which has been popular has been the invention of hard parts, but as I've tried to explain, that makes good ecological sense in terms of predator-prey interaction. I don't think it's a trigger itself. Another one which has attracted a great deal of attention is this so-called snowball Earth, the idea that there were runaway glaciations. This, again, geologically is a fascinating thing to teach, but it's also very controversial. And by no means everybody is persuaded that the Earth became completely glaciated. But even if it did, and even if thereby produced a series of biological bottlenecks, and only the amelioration of the environment allowed further diversification of life, including animals, the problem with these snowball Earths is that they significantly predate the Cambrian explosion. Cause and effect simply seem to be too far apart. But even so, that may turn out to need further study. Yet another idea has to do with oxygen. It's rather difficult to find an interesting slide of oxygen, so I thought I'd show you the guy who more or less discovered it. I mentioned this man who fascinates me in many ways, partly because he was produced by somebody who writes a great deal of popular science and should have known better than he doesn't. Antoine Lavoisier was a totally extraordinary individual. Those of you who are Americans can actually take a hat off to him, because he was a person who reformed the gunpowder industry in France, and if he hadn't, it was not easy making industrial quantity gunpowder. If he hadn't, then probably it would not have been sufficient supplies sent to the to the it's coming slowly. He's also well known, of course, because he was a victim of the terror. And this is said to be a picture of him shortly before his execution. And again, it's worth mentioning, I think he went to his death with exemplary courage, totally exemplary. Invention of eyes. Well, this is an idea put forward by Andrew Parker. Yes. And again, I mean, this is sort of the classic sort of cause and effect problem. Andrews tries to argue that, trilobites were really the key to all this. The only trouble is that trilobites appeared long after the beginning of the Cambrian explosion, and so we go on. And also, as I'm trying to explain with regard to the evolution of eyes, they're convergent, but in addition, they're gradual. You don't just get eyes popping out of nowhere. And there I would agree entirely with Dawkins. A 1% eye is a whole lot better than a 0% eye. So, yes, eyes are part of the story, but again, the sensory systems we see in other contexts are part of the story, but I just don't think it's likely to be the answer to the Cambrian explosion. And so far as there is an explanation, my own money would be, actually I'm rather sort of schizophrenic about this, my own money is probably largely on changes in atmospheric oxygen. But other people are becoming very interested in some of the molecular construction and possibly some of the molecular innovations which separate a sponge from the more complex animals. And they're especially interested in the evolution of the area to do with the digestive system. And that may, in fact, be the most fertile area to look at. And then just to conclude, of course, this is really going back on old territory. In addition to the Darwin's dilemma, of course, the Burgess Shale attracted a huge amount of attention, courtesy of Stephen Jay Gould. But the reason why he wrote his book, of course, was just to keep his ideology up and running, and that was, of course, the rerunning of the Tape of Life. And I was addressing that in the previous lecture, so I don't need to return to that. But of course, just again to remind you that I think the evidence for directionality in evolution, as exemplified by the convergence between the Lophotrochozone and the Deuterostome, is just one of many lines of evidence that suggests that the idea that rerunning the tape would produce a completely different biosphere is less likely to be correct. But behind all that, in a little bit, it goes in part of a way to try and answer Dennis's question about early extrasolar systems. And this is not work by me, it's far beyond my competence. But there are a few straws in the wind now to suggest that even though there may be many Earth-like planets, in point of fact, they themselves may have radically different histories. And in particular, there are a number of things which are of interest. This is a work done by the Potsdam group, but what's shown is, I think you can read, is surface temperature from the past to the present day to the future. And what's becoming fairly clear is that, perhaps consistent with the idea that evolution was difficult for much of Earth history, was because of the elevated surface temperatures. Eukaryotes generally find it pretty intolerable above 45 degrees Celsius, give or take. It's not a comfortable time for them. Prokaryotes are fine. They can live in 121. They can live in boiling water. But what's more striking is, first of all, we don't really understand why we have the curve we do. It's broadly to do with feedback mechanisms, especially through what we call carbonate-silicate weathering and also the evolution of plants. But note well that on this model, and there are other lines of evidence to support this, in point of fact, our planet becomes almost uninhabitable in less than 1 billion years. Also, in terms of the history of this planet, there is some suggestions now that the oxygenation per se was by no means inevitable. There's also evidence to suggest that there are runaway snowball episodes, if not in the Neo-Proterozoic, then in the Paleo-Proterozoic. So there is a possibility that, in fact, the lines of history which different Earth-like planets might go along may be actually more different than we would expect. And that might explain that even though things are, quote, inevitable, the suitable conditions on the surface for evolution may be more problematic than has been thought. This is very, very uncertain how these arguments may go. And that, I think, is a fairly sort of sobering thought at the moment. And then the other aspect we say is we can begin to think what extraterrestrial planets would look like. I just thought I'd show you to finish. This is fun we had with a television program. This is a tightly locked planet, Earth-like, rotating around an M-star. M-stars are not places to hang around because though they're very long-lived, they have intense ultraviolet activity, so you have to evolve that. But because the sunlight's always beating one point there, because the sun never changes its position in the sky, you have this super hurricane here. So you can have a lot of fun working out what it would be like living there, or on the frozen side on the back there. So whether we get the chance, though I wouldn't be surprised, of imaging some of these things remains to be seen. But whether they each had their Cambrian explosion is something which at the moment will be quiet about. So I'll finish there. Thank you very much again.
Thank you. Well, we just have a few minutes before we exercise our legs and walk all the way home to dinner. And Ruth's got a microphone, so any questions?
So Simon, that was a very interesting set of new data that I wasn't aware of in terms of new discoveries about what's going on in the fossil record. In Science magazine a couple of months ago, the sequence of the sea anemone was published. This is Cnidarian, and this is a very interesting result to many of us because it was quite surprising that the genome sequence of the sea anemone is actually closer to vertebrates than expected, substantially so, because we've depended so much on the comparisons with Drosophila and C. elegans as organisms that we figured were giving us a glimpse further back in evolution. And yet here we have the sea anemone, which I guess I have understood common ancestor with that and us is perhaps 700 million years back. And the menu of genes that the sea anemone has to work with is much more similar to ours than people would have expected. Not only the genes, but actually even in some cases the order of the genes, the synteny, has been preserved over a very long period of time. That suggests, just given what apparently the common ancestor had to work with, the eumetazoan ancestor, that at least in terms of capabilities of generating lots of body plan complexity, there was a lot there, a lot more than I think many people had appreciated. How do you fit that into the fossil discoveries, or is it possible to do so, or do we just need many more genomes of other organisms in that same part of the evolutionary tree to make sense of this?
There's a straw in the wind. I know of the paper. I've not read it, I confess. I'm sort of snowed with literature, but I know of the work, and I know a little bit about the related stuff on the anemone stuff, but I can't speak with authority, is what I'm trying to say, if I could on anything. The straw in the wind is that amongst the, and I'm not sure whether this will be widely accepted, within the deuterostomes there is a weird creature called Xenotabella. Very odd, very simplified thing, which may be related to the echinoderms plus hemichordates. Xenotabella has a nervous system which is not so dissimilar to the acoelomorphs, which are one group of platyhelminths which are not derived. But the acoelomorphs themselves do have echoes of Cnidarian organisation in them. So this again is making some big jumps between what are still quite disparate body forms. But I wouldn't be too phased, and there is other molecular evidence that suggests that actually deuterostomes, even though they produce things as interesting as vertebrates, are actually surprisingly primitive. They are further down than the ecdysozoans or the lophotrochozoans. The other thing, though, and I hesitate to even mention this, given I'm dealing in your area of expertise, and it's very far removed from what I even dimly understand. But to the first approximation, what, of course, was very striking, I think, in the genome story, was just how small the genomes are in comparison with protistans. And in the case of the three major groups, all of them are stripped down to around about 20,000 genes. And that may be very important, the sorts of things you were referring to with regard to disease and so forth. But in fact, if I understand it correctly, and of course it all depends what you mean by gene, that's another question, but you know, if a diatom's got 100,000 genes and we've got 25,000, say, or whatever, and yet a diatom doesn't speak, what's going on? Well, I mean, you know, you can't build a complex form with cognition using that number of genes. You'd never get there, it just take too long. You just have to strip yourself down. So there's a convergence in the stripping down of the genomes of these advanced groups, which actually has a parallel with pathogenic bacteria. You know, and these other, you know, these symbiotic bacteria, when you strip them right down, you export most of your genes, and that for a slightly different reason, of course. And I think what's fascinating about this is, you know, to ask, you know, what is the molecular organisation which allows you to make big, complex bodies quickly and efficiently? And in that sort of way, of course, what's so fascinating, though I think actually unsurprising, is that people, so again, have this sort of intuitive idea, you see an enemy, sort of squash it, you know, it's primitive. These things are incredibly complicated, and in some ways more complicated from one perspective than we are. But I don't have any, I mean, I don't have any particular problem with what those connections might be. But again, what's fascinating from the evolutionary viewpoint, and I'm sorry I don't know the paper in detail, is those clear molecular footprints which take you, you know, say from Nematostella to Amphioxus. It's remarkable, yes.
More questions?
Simon, I was wondering if you come across fossils of developmental intermediate forms of arthropods or other animals.
Not really, no. I mean, there are those embryos, and there's another embryo story which has been taken forward very brilliantly by a guy called Phil Donoghue in Bristol. He's done some brilliant work on the Cambrian equivalents. But those effectively, they're direct development. So they don't go through the catastrophic metamorphosis, which I know we were having a conversation over tea. Crudely, in marine life, you can either go through direct development, where the egg more or less transforms itself into the adult form without too much fuss and bother, or you could turn yourself into a feeding larvae, which is often up in the water column, and then the whole thing falls apart, so it basically blows up, and the completely new thing comes out of the preceding thing. The direct development is probably primitive, but no, most of that you don't get the sort of useful encoding. And oddly enough, in the Burgess Shale, and even I think Chengjiang, there are very few juveniles. They're very, you know, we can, you know, obviously they're often smudge-like, but once you get your eye, you can easily recognise things a millimetre across. It's not very difficult, and there's not much of that around. So, but most, because it's marine stuff, obviously we've got fish. We don't know anything about their embryology. There's stuff to do with hagfish and all the rest of it. Most of those, I think, showed direct development, and in a certain sort of way were less interesting, if you like, than the ones which go through the indirect development and the catastrophic, and with so-called set-aside cells and all that stuff.
Our microphone area would like to have a question.
The fact of the explosion, as a biologist, just makes me want to start thinking about epigenetic changes. Is that a big topic of discussion in this area, or is it?
Not really, no. I mean, I'm sorry, I was verbal, I need to drink. But there are so many areas of expertise which one's got to put one's toe into. Some years ago, there was a huge amount of interest, of course, in homeotic genes, and the general idea that you could more or less remake a body plan as you like, but in a way which may be less sympathetic to the genetic side of an audience. You know, in a funny sort of way, they're not terribly important. You know, they do things. Yeah, they run things. You know, they do things in the right order. But it's not clear to me the extent to which the genes determine the morphology, or as my friend and colleague Graham Budd would argue, actually the morphology turned the genes. So in fact, the genes spent their entire time just trying to keep up with organising your body. So of course you've got to have an effective genomic system because you've got to get through your embryology very quickly. So naturally the whole thing's streamed down, all organised, all the ducks in a row and this sort of business. But the sort of epigenetics of this and in one sense it's fine and large. I don't have any quarrel with the sort of ease of transformation between these different stages. I think can be put in a genomic context. I don't have any problem with that at all. What one would like to know, though, is why sometimes gene duplications appear to be extremely important, and many times appear to be completely trivial. And they may certainly be, sometimes they're useful, sometimes they're not. There isn't any general theory about that. But as I'm sort of trying to indicate, in the naive belief we're looking for some general theories in biology, those would be the sorts of things to investigate. So Nigel Hughes has done some very nice work, effectively, on trilobites, looking at their sort of homeotic patterns. And they're sort of quite convincing, they've got lots of pretty colours, and Hox 10 or whatever it is sitting there and all the like. But to date, I don't really think I've come across a great deal which is more than storytelling. It's perfectly convincing storytelling, but it's using solely what we know about extant genomic systems and importing them onto fossil forms. And again, I've no quarrel with that. It may produce some interesting questions, but I just don't see the sort of excitement that, in the way Rudy Raff was writing about in particular in his Shape of Life book, has not quite materialised yet.
Thank you. Can I just ask a quick one which you both might be able to answer? We heard about getting DNA from Neanderthals and from fossilized mammoths and things. How far back is it likely we can actually get a sample from some old fossils so we can actually get some connection between the fossil record and the genomic record?
I'd say not good.
It's really tough. I mean, the Neanderthal experience.
Could use the mic.
Oh, sure.
Yes.
So the Neanderthal experience has been instructive in this regard. Before that, there was a good deal of effort on cave bears that were about of the same age, but where the fossils were less likely to be contaminated by human fiddling. And again, that's the big issue, is contamination. If you're sequencing a cave bear and you get a sequence that looks like human, you know it wasn't the cave bear. If you're sequencing the Neanderthal and you get 25 nucleotides that look a lot like Homo sapiens, well, but maybe there's one change. Well, is that actually a human DNA sample, or is it Neanderthal? And of course, the problem is there is chemical degradation of DNA that's been sitting there for a long time in a predictable way, but still it's very difficult to reconstruct what the sequence might be because of those predictable deaminations. I don't know that I'd want to say never, but it looks like it will be extremely difficult to go beyond, say, 100,000 years and be able to get readable DNA sequence, which means an awful lot. And the things we're interested in are out of range now. Maybe I'll be...
No, I agree with that. I mean, just cap it to say two things perhaps. I mean, first of all, the excitement with the amber and also some of the fossil plant has turned out, to date, there may yet be recoverable. It's been certainly a disappointment. And that's perfectly understandable. I know that people did it in good faith, if you like, and they just didn't realise how stringent the protocols had to be. But I think there's a slightly different aspect also, which goes back almost to the question about the epigenetics and the way in which evolution works. And we do know that these developmental systems are convergent in their own way, and that they are sort of very reactive. And there's a fascinating paper, for instance, on a particular group of plants which involve water storage structures, these ones which live in East Africa and Madagascar. And as the investigator points out, first of all, probably to make particular complex structures, genomically is trivial. The changes you need to go from one sort to another is very, very small. It may be quite sensitive, and that's fine, it may be environmentally sensitive. But equally importantly, this is something which is a take-home message sometimes for some of our anti-Darwinian friends, is that you don't necessarily have to have missing links. You don't have to have an automatic gradation which takes you from stage A to stage 3 through stage B. You can actually flicker in and out. These morphological organisations can appear like that, they're there. You change 2 amino acid sites in one place, changes the other direction. The effect is totally profound, but the actual genomic differences are completely trivial. So that's really, I think, that's why I have such trouble with the epigenetic thing. It's actually pinpointing what one's trying to find.
Yeah,
I've no quarrel with it, it's just very difficult.
The stickleback work is
Yes,
very
of course.
instructive.
That's right, exactly, yes.